So, erythrocytes, also known as red blood cells, are the most common blood cell. Males have about 5.4 million red blood cells per microliter of blood, and females have about 4.8 million per microliter of blood. And red blood cells or erythrocytes actually make up about 25% of the total cells in the body. And they're really small with a mean diameter of about 8 micrometers. And they have no nucleus and they look like a dinner plate. Their main function is to pick up inhaled oxygen from the lungs and transport it to the body tissues. And in turn, they pick up some carbon dioxide, which is a byproduct of cellular respiration, and they transport it back to the lungs for exhalation. So, although leukocytes or white blood cells typically leave the blood cells to perform defensive function, movement of erythrocytes is from the blood vessels is actually abnormal. So these cells don't really leave the blood vessels at all. But white blood cells, on the other hand, are capable of leaving the bloodstream to perform their defensive function. So if you look at the structure of red blood cells, they have a biconcave disc shape with very shallow center, and they have no nucleus in them. So as they mature, they lose their nucleus to accommodate hemoglobin. So insufficient RBCs can cause death in minutes due to a lack of oxygen to the tissues. So oxygen deprivation or ischemia can cause death. So if you look at the shape of red blood cells over here, this is a scanning electron microscopy image of a red blood cell. So they have a shallow center. The cross section makes them look like a dumbbell-shaped cell. And if you look at the blood vessels, these are red blood cells that flow through the bloodstream. So each hemoglobin molecule contains ion, which can bind to oxygen molecules, and each hemoglobin can bind to up to 4 oxygen molecules. They don't have a nucleus. They don't have other organelles like mitochondria to produce energy, and they are biconcave disc-shaped structures. And the reason they don't have a nucleus is just so they can allow the hemoglobin to occupy all the space to efficiently carry oxygen. So each blood cell contains hemoglobin. Hemoglobin is the responsible protein for carrying gases and nutrients in the bloodstream. So each hemoglobin molecule contains 4 globin proteins, and these over here are globin proteins, and they contain alpha and beta chains. So they contain, this over here is beta chain 1, beta chain 2, alpha chain 1, and alpha chain 2. And the center of each globin chain, you see a red pigment, which is the heme group. And the heme group contains an ion of iron that is responsible for carrying oxygen and carbon dioxide. So every molecule of hemoglobin contains 4 globin proteins with 1 heme pigment each. And each erythrocyte or each red blood cell can contain up to 300 million hemoglobin molecules. And therefore, each of these can transport almost up to 1 billion oxygen molecules. So each heme group or each hemoglobin can actually bind to up to 4 oxygen molecules at the center of the heme group that you see over here. Because each of these ions can bind to one oxygen molecule. And individual erythrocytes can bind to about 1 billion or 1.2 billion oxygen molecules. Okay, so red blood cells, they contain hemoglobin for regulating the blood flow and the blood pressure. They also release nitric oxide, which is a potent vasodilator. So they improve blood flow because they help in vasodilation. And they also contain carbonic anhydrase, which is an enzyme that is responsible for conversion of carbon dioxide and water to carbonic acid. And the carbonic acid is the main buffer in the blood, which helps in maintaining the pH of the blood. The normal pH of the blood is pH 7.35 to 7.45. Anything below 7.35 makes the blood acidic. Anything above 7.45 makes the blood basic or alkaline in nature. So the red blood cells transport about 70% of the carbon dioxide in plasma. Okay. So erythropoiesis is the production of red blood cells, and it begins in the bone marrow. And when the red blood cells are immature, they're called reticulocytes, which are immature RBCs. They enter the circulation and they mature in about a day or two. And as they grow, they'll lose all the cell organelles to accommodate hemoglobin. And a mature red blood cell lives in the system for about 100-120 days, after which they are destroyed. So erythropoietin is the hormone released by the kidneys when the concentration of oxygen is really, really low. And this stimulates the differentiation of hematopoietic stem cells to generate more red blood cells. So when the oxygen level is low, the stem cells, they start differentiating into erythrocytes, and the first cell they form are erythroblasts. The erythroblasts further convert to reticulocytes, which is right over here. These are immature red blood cells. Immature red blood cells are released into the bloodstream where they mature, and they circulate for an average of 120 days. And old and damaged cells are then phagocytosed by macrophages. Macrophages are phagocytic cells that break up the old damaged red blood cells. And this happens mainly in the bone marrow, the liver, and spleen. And the globin, or the protein portion of the hemoglobin, is then metabolized or broken down into amino acids, and they're again reused for protein synthesis later. So the heme portion is broken down into a component known as biliverdin, and the ions of iron are bound to a protein known as transferrin. And both bilirubin and transferrin are then taken to the liver, and unused heme groups are then recycled and used in synthesis of red blood cells again. And bilirubin again is used in hemopoiesis, but and some of it is lost by the body. So the next type of blood cells are leukocytes, which are nothing but white blood cells. The two types of leukocytes, granulocytes and agranulocytes. Now, a special characteristics of white blood cells is that they are capable of leaving the bloodstream or the blood vessel, and they're capable of moving through the connective tissue of the dermis to reach the site of a wound. Some leukocytes like eosinophils and neutrophils are granular leukocytes that release certain chemicals to destroy pathogen. So they're capable of leaving the bloodstream by squeezing through the epithelium, which is a process known as emigration or diapedesis. So yeah, if you look at this picture over here, it shows a process wherein leukocytes are capable of exiting the blood cells and moving through the connective tissue of the dermis to reach the site of wound. Now, the first step in this process is that the leukocytes in the blood respond to chemical attractants that are released by pathogens, and they then, or say injury, so they then squeeze through the epithelial layer, and certain proteins known as integrins or neutrophil allow them to stick to the endothelial layer and then squeeze out of the endothelial cells of the capillary as they follow the chemical signals. And this is called chemotaxis, wherein they migrate towards the chemicals that are secreted by the pathogen. And in the damaged tissue, monocytes, which are other specialized white blood cells, they can differentiate into macrophages. And macrophages are phagocytic in nature. So the macrophages basically eat up pathogen and present it to the immune system. The eosinophils and neutrophils release chemicals that break apart pathogen. So this is a very special characteristic which we call diapedesis or emigration. So certain factors known as colony stimulating factor and interleukins prompt bone marrow to generate white blood cells when needed. And in a normal person, the white blood cell count is below 11,000 cells per millimeter cube of blood. Now, when the count of WBCs go beyond 11,000, it is a good indication of an infection. For example, say you have a flu or a mononucleosis infection. The white blood cell count, they shoot beyond 11,000 cells. And when the leukocytes go beyond 11,000, it's a standard indication of an infection. But then they go down once the infection has been controlled. Some other conditions include leukopenia. Leukopenia is a condition wherein a person has very few leukocytes. And if this condition is really prominent, the individual has a really poor ability to fight infections. And the excessive leukocyte proliferation, on the other hand, is called leukocytosis. Although leukocyte counts are high, the cells themselves are often non-functional. So a person with leukopenia is capable of producing excessive amount of white blood cells at the time of infection, but they generally are non-functional, which leaves the person at really high risks for diseases. Then another condition that you probably have heard of is leukemia, which is a cancer that involves overproduction of leukocytes. And this usually involves specific leukocytes either in the myeloid line or the lymphoid line. In chronic leukemia, basically mature leukocytes accumulate and fail to die, which leads to a cancerous condition wherein you have abnormally high number of white blood cells. Now, lymphoma, on the other hand, is a form of cancer which you have excessive amounts of T and B lymphocytes that collect in the lymph node and the spleen. So platelets are the last type of little dust bunny-like structures you find in a blood smear, and these are responsible for blood clotting. So another name for platelets is thrombocytes. THROMBOCYTES. So thrombocytes or platelets are small non-nucleated fragments of a cell known as megakaryocyte. Now, the platelet is not a cell, it's just a fragment of the cytoplasm which contains clotting factors. So these are also descendants of myeloid stem cells, and they are initially, the megakaryocytes are really, really large, about 50 to 100 micrometer in diameter, but then they break down to really tiny granules of about 2 to 4 micrometers, which you call platelets or thrombocytes, and they contain a complex internal structure which you call a canalicular system. So they're capable of moving by amoeboid movement. So they can move by like an amoeba and cause phagocytosis. A normal platelet count in a person is typically 130,000 to 400,000 platelets per microliter of blood. So their main function is to help with blood clotting. So blood clot involves a series of reactions between different types of clotting factors in the presence of fibrin proteins to form a really fibrous structure that we call a clot. So platelets are actually critical to maintaining homeostasis and stopping blood flow in a damaged blood vessel. And they also secrete other things like growth factors, which are important for growth and repair of tissue, specifically connective tissue. So if you look at the homeostatic mechanisms that is performed by platelets, it usually happens in 3 steps. The first one is vascular spasm. Vascular spasm involves vasoconstriction, a brief tightening of the muscles to constrict blood flow. And then it involves the next step, which includes making something called as a platelet plug to initiate a blood clot. And the third step is for actually blood coagulation or blood clotting. So the first step is when there's a break. It causes vascular spasm or contraction of the smooth muscles, which prevents excessive flow of blood, and it brings the vessels closer so it can tighten the gap of the breakage. And the next step is to release chemicals known as clotting factors by nearby platelets, and this is initially sticky, so it attracts other clotting factors to initiate the formation of something called as a platelet plug. So the third step is blood coagulation, wherein fibrin proteins specifically form a mesh that traps other red blood cells and platelets, leading to the formation of a clot. So some major disorders of the platelets include thrombocytopenia. Thrombocytopenia is the insufficient number of circulating platelets due to bone marrow suppression. So the diagnosis include the formation of petechiae. Petechiae are small purple spots on the skin, and that usually is accompanied by mucosal bleeding. So they usually perform a complete blood count with platelets, and they look at coagulation factors and a peripheral blood smear for testing for the presence of thrombocytopenia. Now, a lot of patients with thrombocytopenia may have heavy gastrointestinal bleeding. Well, not all of them. Sometimes they can have heavy gastrointestinal bleeding. But usually patients have superficial bleeding following an injury, and this usually leads to insufficient clotting factors and delayed blood clotting. So the other condition of the platelet is hemophilia. Hemophilia is a bleeding disorder wherein the blood usually does not have the clotting factor or coagulation factor, usually clotting factor number 8, which leads to a prolonged bleeding and the inability to clot blood, even from a very, very small injury. So the next part of the lecture is on blood groups and blood type. We know that there are some very common blood groups that you hear of. You hear of type A blood, type B blood, AB blood, or type O blood. And people have a positive or negative blood group depending on the rhesus factor or the absence of rhesus factor. So a person who has both the alleles or forms of genes for positive rhesus factor is called a positive blood type. So a person with O positive blood will have a positive rhesus factor, and a person with negative blood type will have both the alleles or genes for a negative Rh factor. So blood group antigens are basically surface marker on the type of the blood cells. So a person with type A blood has type A antigens on the surface. A person with type B blood has type B antigens on the surface. A person with type AB antigen has both A and B antigens on the surface. And if you look at the actual chemical structure of the antigens on the surface, you see here type A has, O, A, and B, all of them have the same carbohydrate chain up to a certain limit over here. And you can see fructose in all of them. And you can see, one galactose group, one glucose group, another galactose ring, and then you have another glucose and N-acetylglucosamine ring in all the antigens. But if you see the last side chain in O, A, and B, type A has N-acetylglucosamine in the corner along with the normal common carbohydrate chain. Type B blood had a galactose instead of N-acetylglucosamine. Now type O blood has neither N-acetylglucosamine nor galactose on the surface. So that makes type O different from type A and type B blood. So the presence of different antigens on the surface makes the blood group really, really different. So the rhesus factor is basically determined by the genes on chromosome 1, which we call the rhesus group. Now some people have a positive blood type and some people have a negative blood type based on the rhesus factor. Now we have 2 alleles for rhesus factor on each chromosome. So in simple terms, this is an inherited protein that you find on the surface of blood cells, If your blood cell expresses this protein, you're Rh positive. If your blood cell does not express this protein, you're Rh negative. And that's how we get a positive and negative blood type. Now, rhesus factor is very important in women who have a negative blood type. So a woman who has a negative blood type, if she has a baby which ends up having a positive blood type, during the third trimester, she produces antibodies against the positive blood type in the baby. This is not a problem during the first pregnancy because this doesn't happen until the third trimester. But in the second baby, it causes a condition known as hemolytic disease of the fetus and newborn, because of which she starts to reject the second baby and it causes miscarriage. Now, in rare conditions where, say, the woman has had a blood transfusion with the wrong blood type, she can have a miscarriage even in the first child, or it can also lead to HDFN in the first child. So to avoid HDFN, which causes hemolytic disease in the newborn and it causes cyanosis in the newborn and makes the baby born blue, they inject a pregnant woman with RhoGAM. RhoGAM basically is administered at about 28 weeks of pregnancy, and again 72 hours before delivery. And this basically prevents HDFN in babies who have a mother that have a negative blood type. So blood types can be of different types: O, A, B, AB, and Rh. There's also another blood type that you call a Bombay blood group. It's a very, very rare group that lacks the normal antigens of ABO. So a person with Bombay blood group can neither receive or take blood from ABO blood type, but they can only receive blood from a Bombay blood group type. It's a very, very, very rare type of blood group. Now, the most common type of blood is usually positive blood. It can be O, A, B, and AB. AB in general is the rarest form of blood type, and O is very common in Native American descent, European American descent. It's relatively less common in Korean American and Japanese American people in the United States. Now, this chart over here shows the blood group type and the types of antigen they have on their surface. Now, when they have the antigen on the surface, they have the opposing antibodies in the plasma. For example, type A blood have type A antigens on the surface, but they'll have anti-B antibodies in the plasma. Because if they have the same antibody, these antibodies will react with type A antigen and cause agglutination or precipitation of the antigens in the blood. And that's why if you have type A blood, you have the opposing antibodies. Anti-B means antibody type B. And if you have type B blood, you have type B antigens on the surface and antibody A in the blood plasma. Okay. Now, a person with type O blood, keep in mind they don't have either A or B antigen on the surface. So in the blood plasma, they have both anti-A and anti-B antibodies. And a person with type AB blood has neither type A nor type B antibodies in the plasma. So these guys can receive blood from A, B blood group, or AB or O. So type A, type B, type AB or O. So this type of blood group is called a universal recipient. Now, type O blood is called the universal donor. So usually when a person needs an immediate transfusion, they can start a blood transfusion with type O blood, and then they wait for the blood report to see what type of blood they need to continue with the blood transfusion. But this type O blood is called a universal donor. So blood typing is performed usually with anti-serum, which are nothing but antibodies against a particular antigen that are synthesized in a bunny rabbit. So that they take type A blood with type A antigens on it, and they inject those antigens in a bunny rabbit. The bunny rabbit produces antibodies against antigen A, and they pack those antibodies in a bottle to be used for blood testing. Keep in mind, type A antigen will react with type A antibodies. All right. Similarly, type B antibodies are produced by injecting type B antigens in a bunny rabbit. The bunny rabbit produces antibodies against type B blood. So in the Kitson lab, you'll have anti-serum A, anti-serum B, and anti-D. Anti-D is the antibodies produced against rhesus factor. So that you don't see over here, but just keep that in mind. Anti-D is the antibodies produced against rhesus factor, positive rhesus factor. So here you can see that a person with type A blood, their blood will react with anti-A serum. A person with type B blood, their blood will react with anti-B serum. A person who has type AB blood has antigen A and B on the surface. So the blood will react or agglutinate. This process over here is called agglutination. So the blood will agglutinate with both anti-A and anti-B people. Now, type O blood does not have antigen A or B on the surface, so you don't see any reaction at all. So this over here is untreated blood, which is just a control to make sure you're doing the experiment right. So a very important condition of the blood is sickle cell anemia. Sickle cell anemia is a very serious condition where there's a mutation in the blood. A normal blood has glutamic acid in the protein sequence, but a person with sickle cell anemia, instead of they have a small mutation, so the RNA sequence, instead of GAG, it ends up having GUG, which leads to valine instead of glutamic acid. So this small change in the amino acid leads to an abnormal shaped red blood cell, which looks like sickle shaped cells in the blood. And a person with sickle cell anemia does not get infected with malaria. So you see a lot of sickle cell anemia carriers or infected people in regions of the world that have a lot of malarial infections. So it's a blessing in disguise because they do not get infected with malaria. However, a person who is actually infected with sickle cell anemia, they start having clumps of sickle cells blood that block the blood flow, and it leads to pain and serious infection and sometimes organ damage. So usually sickle cell blockage of the blood vessel is accompanied by pain and fever. It goes away about in say 15 days. If it's in the hands and legs, it's not a problem. But imagine having blockage of blood flow in the heart or the brain. So people who end up having a blockage due to sickle cell anemia in the heart or blood can die. So they have a very low lifespan if they actually get affected. But not everybody who has a gene gets affected. You need both the alleles for sickle cell anemia to actually be infected. So a person who is a carrier can have some amount of sickle cell blood cells, but not a whole lot of them. So these are some important things that you need to know about blood and some important condition of the blood. But some other conditions that you probably have heard of of is hemorrhagic anemia, which is due to, well, anemia, different types of anemia, like hemorrhagic anemia, which causes sudden hemorrhage in the blood. Then you have pernicious anemia, which is due to the lack of vitamin B12, which is usually as a result of intrinsic factor, which prevents absorption of the vitamin in the small intestine. And then you have hemolytic anemia, which leads to excessive lysis of red blood cells usually due to bacterial infection. And then a very common condition is iron deficiency anemia. And iron deficiency anemia is due to the lack of iron in the diet, or depletion of iron reserves in the blood, which causes the blood to be really thin, and the person looks pale because of lack of hemoglobin. And then a condition we looked at is sickle cell anemia, wherein this is a genetic defect which leads to sickle-shaped red blood cells, which leads to poor transport of oxygen through the body. And this type of anemia is very common in people with African descent, especially in people who live in malaria-prone regions. These are some things that you need to know in blood, well, in this chapter. So overall, you need to know to describe the properties of blood, the composition of blood, different types of blood cells, and in general, how blood helps in maintaining homeostasis. Know the major conditions or disorders of blood, which is caused by maybe disorders due to erythrocytes, white blood cells, and platelets. So I'm sure that in lab you'll study a lot more about different type of blood cells that you can see in a blood smear, and certain clinical conditions that can be diagnosed by looking at a blood smear.